Biology Crash Course by Juliana Castrillón, Daniela Arbeláez, Paula Garcia, and Sofia Valencia. This crash course will help you study for the state exam. First, we'll talk about biomolecules, nucleic acids, protein synthesis, and finally, mutations. First, biomolecules. Carbohydrates are organic compounds made of carbon, hydrogen, and oxygen atoms. Carbs are made of monosaccharides linked together. Their function is to provide a key source of energy for cells. Lipids are nonpolar molecules that are not soluble in water. They include fats, phospholipids, steroids, and waxes. Its function is to provide energy and serve an important part in the structure and functioning of cell membranes. There are also proteins, which are chains of amino acids. They can be enzymes to promote chemical reactions, or they can help structure your ligaments, tendons, bones, hair, and muscles. Proteins, called antibodies, help your body defend against infection. Finally, there's nucleic acids, long chains of smaller molecules called nucleotides. Nucleic acids mainly serve the purpose of providing the organism with its genetic blueprint and coding. They basically tell you who you are. This type of biogeochemical molecule only brings us to the next topic, which are nucleic acids, more into DNA and RNA. DNA carries the genetic information in the cell, and it's capable of self-replication and synthesis of RNA. It consists of two long chains of nucleotides twisted into a double helix and joined by hydrogen bonds, which I'll talk about later. So the structure goes like this. The two red parts of the DNA, which are twisted, are the sugar-phosphate backbones. Then, there's the nitrogenous bases, which are the little colored sticks. Finally, there's the gene, which is a complete twist of the helix. This is assuming of how the molecule actually looks like. The blue circles are the phosphates, which are organized as 5' for the first phosphate row and 3' for the last. These bond with the sugar, in DNA called deoxyribose, and finally, they bond with the nitrogenous base. This process is repeated in a mirror-like action, whereas the first nitrogenous base bonds with another, followed by a sugar and then a phosphate. These bases are called adenine, guanine, cytosine, and thymine, but we will call them only by their initials. The next nucleic acid is RNA, which is single-stranded. RNA molecules are involved in protein synthesis and sometimes in the transmission of genetic information. Its structure, as I said before, is based on just one strand. In the zoom-in, we can see only one phosphate row and only one sugar row. This means there's only one strand. As for the nitrogenous bases, there's adenine, guanine, uracil, and cytosine. There's no thymine anymore. But what about the specific bonds the nitrogenous bases have to adapt to? In DNA, C can only bond with G, as well as A only bonds with T, and vice versa. In RNA, C only bonds with G, and A only bonds with U, and vice versa. There are many differences between DNA and RNA. For example, DNA is double helix and RNA is one-stranded. There's also thymine in DNA, and RNA has uracil instead. And also DNA is a blueprint of biological guidance, while RNA only carries that blueprint. We come to the most complex topic of all, protein synthesis. It has two main steps, transcription and translation. Transcription actually happens in the nucleus of the cell. In transcription, we'll talk about mRNA, the red string in the picture. It will travel through DNA and copy the information needed for protein. Then it will go to the ribosome. In the ribosome, the mRNA will hold the basic information and tRNA will carry the amino acids needed to complete the protein and form a polypeptide chain in a process called translation. When all the amino acids are translated, this will form a polypeptide chain. To understand these steps, we need to know what a codon is. A codon is a group of 3 nucleotides in mRNA, and there's also anticodons, which are groups of 3 nucleotides in tRNA, meaning anticodons bond with codons to form polypeptide chains. Here's an example of the nucleotides you can find in mRNA, where GUG form a codon. In tRNA, CAC form an anticodon. This would be the results of both steps. The groups you can see in the protein line mean those are the amino acids that will be created to form that protein. Now that we know this, we'll talk about mutations, which are a change of the DNA sequence within a gene or a chromosome of an organism, resulting in the creation of a new character or trait not found in the parental type. Mutations can be classified by either their effect on DNA or on protein. There's missense and nonsense mutations, which are classified by their effects on DNA. And here's an example of point mutations. We can see that if one nucleotide changes in DNA, it will change RNA's result and finally obtain a different nucleotide. As for the frameshift mutations, if one is added to the DNA sequence, it will change the codon groups in RNA, and practically it will change almost every amino acid result in protein. Now the big question, is mutation a disease? It actually isn't. If mutations are silent, meaning they won't change the final result, they do not affect a person at all, but if they do change, they can either affect one's health or not. The thing is, the result can actually make people more vulnerable and they may get affected in health more easily. Thank you for watching. We hope it helped.